Give you my perspective. During my general surgery training at UCSF, I created a lot of holes, took out a lot of cancer, took out a lot of disease, and created a lot of massive, or beyond cellular, but whole organ or tissue deficits. Then on the other side of the table, after doing plastic surgery training in New York, I was responsible for reconstructing or filling those holes. And then ultimately working on children's faces as a pediatric plastic surgeon, I've become obsessed with scarring. So my take on this is a little different. I just point this out. This was an article that Lawrence Baker of the School of Medicine, who's a health economist, and Hank Greeley from law school and I wrote. It's outdated now, but this was about 2 years just after the proposition was successfully passed. We wanted to give an example of how you might have a return on this. And this, I think, the spirit of how fast we're going to bring things to the market, how fast we're going to impact lives in California and beyond is important since we're several years into a decade-long process. One of the things that we talked about, and I think is important here, as you've heard from both Dr. Goldstein and Dr. Conklin, is new disease models, the ability to test drugs and fast forward a process that takes $700 or $800 million in 10 years is going to be dramatic. And it'll be novel for these human diseases. The end of the spectrum is growing a new organ, and that's going to take longer. But sometimes in between, we may be able to develop functional tissue that I think is appropriate. And that's a little bit what I'll talk about here. When people talk about stem cell biology and regenerative medicine, the regenerative medicine is less clear. I mean, someone sent this to me the other day, and I said, okay, so let's give an example of some regenerative processes that we might be able to unlock here. First of all, not all fat is bad. Being a plastic surgeon, I can comment on this literally. You should know that despite the epidemic of obesity in children and adults, sadly, in the country, I think it's an incredible natural resource. I'm from the Midwest, and if you walk through where I'm from, there's an abundance of it, and it turns out that between 400,000 and 500,000 Americans paid cash money to have their fat removed. The average one of those liposuction experiments may be 2 or 3 liters. So it turns out that many people have begun to say, is there something in that liquid gold, if you will, that we could use? And it turns out that many labs around the country and around the world have been looking at the smooth muscle type cells that Dr. Conklin talked about that are in the blood vessels in the fat, not the goblets of lipid. So these are stromal cells. Dr. Goldstein talked about the middle layer of the sandwich. This would be the mesodermal, so muscle, bone, cartilage, not the outer inner layers. So it turns out that if you do a digest, and each of those liters contains about 1 billion cells that are mesodermal-like, meaning the middle layer, and they're capable. What you've been able to do, not just in my lab, but in many institutions around the state and around the world for that matter, is to put chemicals, not genes, but chemical coaching, if you will, to take those cells and begin to make them into other types of cells. And these are the types of cells you can make them into. These are not the answer for nerve. These are not the answer for diabetes. But if you need muscle, bone, cartilage, or fat, or muscle, for example, these cells are capable of differentiating in those cell types. What's really interesting is where I come in the equation is after you remove a massive amount of someone's skeleton, for example, might we be able to identify a readily available source of cells today on that patient in the operating room where you could do a very quick lipoaspiration, obtain some fat, identify, and here's what's different. This is a chicken soup of cells. I don't know if there are 5 types of cells, 4 types, or 10 types of cells in that pellet of cells we get from there, but some of them are capable of going to bone. So the question would be if you could identify those types of cells, put them in an environment or niche, create a microenvironment for those cells that drive them down an osteogenic bone-forming pathway, you could then put them back into that patient, or those patient cells, on the operating table. The concept here would be, let's say, for example, you remove part of someone's femur, the long bone in the thigh. You need that to walk around. So from a bioengineering standpoint, and there are great bioengineering departments in this state for sure, whether you're in San Diego or San Francisco or anywhere above and below that, the bioengineers can put a construct in there that is going to be degradable over 2 or 3 years. That's strong enough for you to walk on. but will also potentially be releasing the coaching to turn those fat-derived or bone marrow-derived. I recognize bone marrow has the same population of cells, it's just more painful to get and it's less abundant than fat. This is also a renewable source. This is a green source of cells, if you will. If you keep eating, when you go out to that snack tray, you're making an investment in your health. Just remember that. So the concept would be, you'd be able to take an existing population of tissue cells, as Dr. Goldstein talked about, and then put them back in the patient and drive them down to repair, regenerate, and replace the bone eventually by your fat-derived cells. So there's an example of a tissue-specific cell, as Dr. Goldstein referred to. Just a little bit about what we've done with this in collaboration with bioengineers at UCLA. We're able to take fat in mice. We're able to take the cells derived from fat, did not sort them, so whatever populations are in there, put them in a scaffold that mimics a little bit like a coral background, so to speak, hydroxyapatite. Put that in a defect in the mouse's skull that would never heal in the life of the animal. And 8 weeks later, we were able to regenerate the skull at the same pace in which bone marrow or bone cells would do it. The reason I'm focusing on skeleton is interesting. We have a population that's not getting younger, a population whose expectations are to be quite active in an age that they live to. And if you look at the New England Journal that came out yesterday, there are 2 articles that talk about if you do MRIs of joints on patients in the Framingham Heart Study, they have an increased incidence of osteoarthritis as a function of time if you just screen for it. And if you operate on joints, for example, arthroscopy, you do no better than just standard care. So another target I haven't talked about is cartilage, which is also something that these cells are capable of differentiating into. Now, will they replace it in a way that integrates and heals? Remember, your cartilage has no blood supply. That's why it doesn't heal, so it has to be taken out if it's injured. So cartilage, bone, muscle, these are increasing needs in our society. If you look at fractures in people who are getting older, over 1.5 million times you have to take a bone graft and put it on a fracture, which tells you it's a little different than putting a cast on it. You're concerned about getting that fracture to heal. Do we need to glue in artificial joints and make them titanium? No. Can we put a bioengineered construct that induces cartilage on the surface because of the coaching that will derive from how these cells are being differentiated spatially and temporally? Can that be replaced over a year or 2 into your own joint? Yes, I think that's possible. Is it possible with these tissue-specific cells? I think so. Would it be more likely if we're able to derive those cells from iPS or embryonic? Probably. But at least the indications are in mice that these cells from fat, and in human, in our experiments on mice that don't have an immune system, are capable of becoming bone. And this has been published and you can look at it. So this is what I call bedside tissue engineering. That is, surgeons have very compliant patients. They're paralyzed, they're asleep. You can derive cells from them. If you don't leave the operating room and you're able to identify, this happens all the time. When I did a lot of trauma surgery at San Francisco General Hospital, when you're a shot in the heart or liver and you're losing tens or hundreds of units of blood, you save that blood, you collect it, you spin it down, you put it back in the patient. So there are lots of examples where we harvest something in the operating room from that patient and put it back in. What I'm saying is maybe this is one of the sources that's readily available in the body today that something could be exploited while we're making advances in either embryonic or iPS type cells. Now, I'm also a pediatric plastic surgeon, so I want to give you another example of what is possible, potentially, in regenerative medicine in the broad area. It has nothing to do with stem cells, but it's the type of thing that when we mention small molecules may be possible. This is the second most common birth defect in the world. This is cleft lip and cleft palate. It's about 1 in 1,000 children. The most common birth defect in the world is club foot, a turning into the foot that you treat with serial casting. So this is a big problem. At least it's a big problem for people who treat this. One of the things that we looked at in the laboratory, postdoc Karen Liu, who brought my lab together with Jerry Crabtree's lab, who's a developmental biologist, and a chemistry lab, was to say, could we regulate protein function, not gene function, but proteins in a precise fashion in the body of the animal? And could we use a small molecule or a drug to do that? Could we do it when and wherever we wanted to, and could we precisely control it? Because if you can do that, It's different than a gene knockout. A gene knockout, you've knocked it out from the time the animal, the first cells are formed, and that's it. This would be something that you control temporally when you wanted to. We chose a GSK3 beta. It's a gene that's very socially active, if you will. It's involved in many processes in the out in the cytoplasm and in the nucleus of the cell. Turns things on and turns things off. So if you disrupt this would be a great candidate to see a problem with. It turned out that Dr. Crabtree's lab was interested in this for different reasons over a long period of time, and he had shown that this system that I'm going to use, you could use in a cell dish in a biochemistry methodology to turn on and off GSK3 beta. Turns out you put a tag on here, and if you don't give drug, the tag makes the GSK3 beta degraded, as if it never had that gene or protein, if you will. So in absence of drug, it mimics a knockout. Well, turns out that the palate, like every organ in the body, forms at precise times. Mice are born at about 19 and a half days, but only between days 12 and a half and 14 and a half does the palate form. The palatal shelves come down from the roof of your mouth. The palate divides the nose from the mouth. This is the tongue, just to be clear. It's like this. So the platal shapes come down. They then make a structure that absorbs water at my wrists. They rotate and fuse in a 48-hour window. So this happens over just a few weeks in the embryo, and it doesn't happen at any other time. So the interesting thing is, if you knock out GSK3 beta with a standard knockout, you get a cleft. You see that? Here's the nose, here's the tongue. There's nothing in the roof of the mouth. The question was, in our system, if you didn't use drug with Dr. Crabtree's construct, this is a little unusual, and I know plastic surgeons ask patients to do a lot of unusual things, but pretend you're standing on your tongue and you're looking up at the roof of the mouth. See these folds, those things that you feel with your tongue, the rugae of the palate. This is the back, this is the back, this is the front. Well, this is a cleft. In other words, a gap in between there. That's a knockout. If we don't give drug to our mice when they're born, they also have a cleft palate. So the system of having this protein tag that destabilizes it, for the embryo, it doesn't know any different. It's got the second most common birth defect in the world. Now the question was, if we gave the drug that Dr. Crabtree's system used, and we gave it to it, we could stabilize this GSK3 beta precisely at a time we want, which is very different from a knockout. When it's knocked out forever. So with this system, what Karen Liu did is she was able to give the drug, small molecule, to mom IV or in the belly. It would then go into the bloodstream, transplacentally, cross into the embryo, and it would stabilize the GSK3 beta. If you don't have that, you have a cleft. But notice, only during the 2 days the palate is forming, we're able to prevent the cleft palate from forming by giving a drug to mom. I should tell you that for 4 years, I was at UCSF in the fetal surgery lab trying to do these heroic operations on lambs and mice and rabbits and primates to treat birth defects surgically, and that's a fantastic program they have there, but this would be a dream come true, so-called preventing a common birth defect. If we tried to do it after or before the palate was forming, it didn't work. In contrast, the sternum, another midline structure, your breastbone, forms between days 15 and 17. If we want to rescue that, we had to give the drug only during the 2 days that was forming in the embryo. So the reason I brought this to your attention, it's an article that was published last year, is just an example of how you might be able to do regenerative medicine in a way that takes advantage of small molecules that you might be able to identify using either embryonic stem cells or induced pluripotent cells that may be able to target something and prevent something happening before you're born. The average child with a cleft lip and cleft palate has multiple operations before they're 18, and just an example of one of the possibilities. So I think with that, I'll stop and answer any questions.
Just on the last example, doesn't that presuppose you know exactly who's going to get the defect?
Very good question, but and I don't mean to trivialize it, but ultrasound is about like a handshake in the United States. Most anatomic abnormalities, including clefts, are diagnosed with prenatal ultrasound. So that really opened up the field 25 years ago by ultrasound to be able to diagnose a problem. The more important question is, and picking up on what you're asking, which is important, is this is an entire jury-rigged system. We know these mice are going to develop clefts because we've done something to a gene. We don't know the gene or genes that cause cleft palate, so we're a long way from doing this in humans. It just shows you that if you do have an ability to regulate a protein target that you might identify with a screen, there are ways you could prevent what is a very expensive and complicated series of operations postnatally. But most anatomic problems like number of digits or a cleft can be diagnosed with ultrasound before birth. In fact, most referrals to pediatric plastic surgeons are from the ultrasound.
But it would be after the point where you could intervene.
Correct. So this is just a proof of concept for being able to regulate a protein. We would have to know the genes that form. We'd have to know that you were still competent to be able to rescue it. It's just the concept of being able to identify targets for genes that cause problems, whether it be diseases or birth defects, is enabled by the technology where you're screening either embryonic or postnatal cells.
Although if it did no harm, it's possible that women in pregnancy could have this drug.
Correct. Dr. Townsend's there are plenty of examples where, for example, if you know something is deficient in the maternal diet, like folic acid, for example, is routinely given to pregnant women since it's not teratogenic, it's not going to harm the fetus, and it might lower the probabilities of a birth defect. So if this small molecule, for example, or drug you were giving was innocuous to mom and had some benefit for the children, then you can do the numbers in terms of the benefit ratio and risk to determine if you'd use that approach. Yes?
Are there any markers that you can identify, maybe even like family genetic history, that would give you a sense that a fetus was in danger of developing a certain birth defect?
Sure. So if a mother and father have a child with a cleft, it substantially increases the risk from 1 in 500 to 1 in 1,000 to way more than that. Mm-hmm. If you have a second child, or if either of the probands, either the parents carry a cleft, or have a cleft, there are ways that you can identify more susceptible populations, for sure. Cleft just being an example.
So two parents, let's say, who decide to get pregnant, if something like this came to pass, you could potentially give a drug prophylactic.
Correct. And if it was innocuous to the mother, right, there are two patients here, the pregnant mom and the fetus, then yes, there would be an example where your risks are so substantial that you might want to be able to use an approach like this. But you'd have to identify the gene or genes and the cause. You'd have to be able to have some, in that case, you'd give it from a very early time point, so you'd always be in that window or before it. I'm not representing we know the genes that cause clefts. I was just trying to point out there are some rescue and regenerative medicine approaches that I think are more likely to be moved forward by identifying targets from stem cell biology. Yes?
Is anyone doing anything now with fat cells in humans, or is this strictly all animal?
That's an interesting question. So there have been companies that have formed to bank adipose-derived cells. So like you have cord blood being banked, for example. So people after having aspirational liposuction, again, that's hundreds of thousands of people a year, can bank that. The second thing is there are examples where in Europe now, where people are aspirating bone marrow and taking chips of bone. One of the reasons I'm focused on bone is we never have enough of it in craniofacial surgery. There are very, very few times when you have too much bone in your body, believe me, except in an osteoporosis type setting, but acutely. And there are reports now, case series coming out of Europe, where they've aspirated fat, combined it with bone chips and bone marrow, and had defects that heal in children who would never heal without the therapy, or who had failed multiple previous attempts to use plastic or metal. So there are clinical applications, not in the United States, that I've seen from this. The attraction for me is that you can get it easily. It has a competent population of cells. I'm sure the bioengineers can identify, and the stem cell biologists can identify a population within fat that is even more robust. And we're looking at strategies right now in the lab that we're halfway through that take the same small molecule strategy, give the drug to the mouse that has the defect and the fat-derived cells in it, and we're seeing that those defects are healing more rapidly. Yes.
What are the, I assume there are some serious rejection issues, but I mean, with bone, say, is it really important that these cells come initially from the individual who's going to get them back?
The bone, the cadaveric bone, has dead cells in it, and will have to be replaced slowly and eroded by your body and the cells that come in. So that takes a very long time to replace that, and it's prone to infection if you have bacteria in your bloodstream, like you would have an artificial heart. So the concept of having your cells to make your bone would eliminate all the problems of that. Yes?
With these fat-derived things you were talking about, do you actually know what the cells are that are responsible for the healing there? Is it some sort of mesenchymal stem cell?
Well, it's presumably a mesenchymal cell. I haven't used the word stem because I'm not certain. It's a heterogeneous population of cells that does the trick. But if we isolated a clonal population of cells in there that we have all the markers for a mesenchymal cell meet the criteria, as Dr. Goldstein said, for STEM, I haven't done that yet, and I'm not aware of that being done. But identifying, that's an interesting question, so then you have to ask if the chicken soup or the minestrone soup of cells from you does it, by identifying a subpopulation of cells and driving them more rapidly with the proper coaching in a three-dimensional construct, is that improved? Probably. The ones I showed you in mice were just the unsorted, if you will.
And are there any other companies that are working with purified populations of mesenchymal stem cells?
Lots. Yeah, I think that's a barroom brawl of intellectual property. And, yeah, but yes, there are lots of them. It's a gigantic and growing market for needs of cartilage and bone. But so there's lots of activity in the area. One of the frustrating things about cartilage is, as I alluded to, there's no blood supply, so it doesn't heal, which makes it really hard as a surgeon who works on wound healing to get a cartilage replacement to integrate in the native cartilage on either side, since it's not going to heal without blood supply. So that's the challenge for meniscus or cushioning cartilage in joints. There's certainly no doubt it's a big need and growing. Yes?
If you had a patient or someone in your family who, say, was getting lipo, would you advise them to make their fat cells?
Me personally?
Not, I mean,
My family's in great shape. Should anyone ask me about that, I'm not sure. I would personally. But I think that's a decision that you have to make. Each person has to make. We get plenty of this brought to the lab from surgery centers, as you can imagine. There's nothing else that's going to be done with it. So from a research standpoint, it's pretty available. It's interesting that if you don't have your cord blood saved, and I don't, might this be an opportunity to save some cells that might be available for something I need? Yeah, sure. So I would do it, but
What are they trying to store your fat?
I don't know. There must enough that they're going concerns. So they're certainly.
I don't mean your fat.
Oh, mine is cheap. I don't know. I don't know the price point on that. Liposuction is certainly not free, but I do have an MBA, so I'm familiar a little bit with the business models of those, and I think it's not inexpensive. It's several $100s would be my guess. Yeah.
So if you're not sure that the active ingredient in the chicken soup, so to speak, is a stem cell, is the research being done in this field? Is it does it qualify for CIRM funding?
Well, I think that I don't want to speak for the California Institute of Regenerative Medicine.
Hasn't any of it?
There are disease team planning grants that might target. I don't know the answer from a specific standpoint that might target musculoskeletal needs, which might use postnatal or adult tissue cells, as Dr. Goldstein mentioned. There are increasing requests for funding announcements that come out on translational. Recent come out, we either address a cell type or a bottleneck. So I think maybe someone from CIRM could comment more readily than I could on that.
I think we're required to fund progenitor or pluripotential stem cells. So there would at least be an issue to think about there, because it requires us to be in that space. But as you say, if it's undefined, and it's a fairly important clinical need, the reviewers and eventually the board would make that decision, I think, and I wouldn't like to predict which way it would go. There would be a number of positives, and there would be some negatives in the whole process. So it's a wait and see. But if it's repair and it's significant, and it relates to pluripotent or progenitor cells, absolutely in our area. So at least on the veranda of possibilities, it's very much.
It takes if it's not pluripotent or progenitor, it takes a 2/3 vote from the peer review group to the board. If it's a vital research opportunity that has high value, that he's identified. So it's not the priority, but with a 2/3 vote from peer review because of high value for our mission to reach patients. And Dr. Trounsend is absolutely right. The board can consider it.
And I must say that while I don't have the data now, I am collaborating with Herb Weissman and other biologists at Stanford who will nail down the question of, in that population of cells that can regenerate bone. I should say that 98% of the bone formed are the cells we put in. I didn't go into that detail, but it's not not the patient mouse cells. It's the human cells that we put in that regenerate the bone. They don't go to cartilage. They don't go to muscle. But that's, remember, the bioengineers are giving us a sandbox, if you will, that is screaming bone, the micro niche, the environment we put the cells in. But we'll know shortly whether there are what we would call skeletal progenitor cells within that population.